Okay, so first up, I don't know if any of you have been to the other lectures, but yeah, hopefully you got a chance to go to some of these ones. They were taken by other, some of like my friends who do different subjects. And if you want any help, you can reach out to any of them, and you can do some like group tutoring with them to start with before the HSC. And a little bit just about me. I graduated last year from Pimble, and I'm now doing science at ANU. These are my pets. So I've got two dogs. They're Border Collie cross German Shepherd, and this is my cat. He loves interrupting my work by sitting on my laptop all the time, which is inconvenient, but it's very cute. So I don't mind that much. And okay, so just an overview of what we'll be doing today. We'll just start off with we'll go through each of the different modules, spend about 25 minutes on each module. We'll mostly just be going through practice questions from previous years of HSC. We'll do sort of multiple choice for a couple, and then a couple of longer answer responses. And I'll just try and get you guys to write down your responses as we're going as well, just to make sure you're really thinking of what's going into each question. We'll also have a couple of breaks in between the modules, so you can, ask questions if you need to, get a drink, whatever you need. And then, yeah, we'll just have a bit of a wrap up at the end. So first off, the syllabus. Hopefully all of you are quite familiar with the syllabus by now. But yeah, so there's just an overview. Module 5 is hereditary, with reproduction, cell replication, DNA and polypeptide synthesis, genetic variation, and inheritance patterns. And then, yeah, you can sort of see module 6 is genetic change, so mutation, biotechnology, genetic technologies. And then module 7, we've got infectious disease, so causes of infectious disease, responses to pathogens, immunity, and then prevention, treatment, and control. Typically, the later questions in the HSC, they've been sort of a combination of module 7 and 8, like sort of the longer, at least for the past 2 years, the longer responses have been module 7 and module 8. So it might be a good idea to sort of think up on, like, study up on them a little bit more, or just sort of think of some longer response questions that they might be able to ask you. They like to combine modules for the longer responses, so they can test a wide range of like skills and stuff. So that would be good. And then, so finishing off with module 8, we've got non-infectious disease and disorders. So homeostasis, and then causes and effects, epidemiology, and then prevention and technologies and disorders. Okay, so we'll just go into a bit of like an overview of the actual exam. So there's 2 sections. The first section is just 20 multiple choice questions. On the paper, it says, it sort of recommends doing, I think, 35 minutes for this section. I would say you probably don't need to spend quite that much time on it. Try and get it done in 30 minutes. And you should probably be able to do it faster than that. And it just means you can get actually into the paper and then come back to the multiple choice at the end if you have time. Section 2, it's just 80 marks of short answer questions. Most questions are between 1 and 5 marks, typically sort of 3 or 4, but yeah, it can range between the whole 1 to 5. And then, yeah, there's also 6 marks. I don't know why I left off 6. There are 6-part questions. But then you'll have 2 to 3 sort of longer type questions, and they're usually 7 to 9 marks. You don't really need to write sort of an essay, like, I don't know if many of you do chemistry, that's sort of expecting sort of longer. But for these types of questions, they're more just wanting you to plan out your response and have like a logical flow of ideas, but maybe a couple of paragraphs, like maybe 2 maximum, but it needs to be sort of detailed understanding of what you're doing for the 9 marks. And yeah, you get 5 minutes of reading time, and then the whole paper is 3 hours. In the reading time, it's a good idea to look through the whole paper, make sure you look at the questions at the end so you have an idea of what you're going to be sort of having to answer when you get to the end of the paper. And then go back, and if you have time in the reading time, which it's not that long, so I don't know how much time you'll have, but you can start working on the multiple choice questions when you, well, like not actually writing anything down, but you can start thinking about them. Yeah, I think that's about it. Okay, so the first question, we'll just get into it. So there's a multiple choice question. I'll just get everyone to have a think about it, and then Emma will hopefully put up a poll and you can answer it.
We'll give you about another 10 seconds just as we get a couple more responses.
Okay, so we've got the poll results up here. It's quite spread out. So I'll just go through them with you. So if you're looking at it, it's asked meiosis as the phase that we're trying to be modeling. So that immediately you're thinking it's not going to be D, because that would be a phase in mitosis. Since you can see how these cells, they've been replicated because you've got sort of 2 strands of the DNA in each of the chromosomes. But the sister chromatids are the ones that are being separated here. So you can see that this would be mitosis, because each of the cells that you get on either side will have exactly the same DNA. So it's not going to be D. You can also see with the option C, there's actually 12 chromosomes there. So in the question's asked for 6 chromosomes. So it can't be C either, which means we're sort of looking at A and B. For option B, the chromosomes aren't lining up in their pairs. So when you have, so I think what they're sort of trying to represent in this phase would be metaphase. But for this, you need sort of the chromosomes to be the same. So this one would have to be sort of like a big one. I can't really draw chromosomes very well, but that would have to be a big one. These 2 would have to be the same size, and these 2 would have to be the same size. So that's not actually a phase that could really occur. So that wouldn't be right. And so it actually ends up being that A is correct. And so you can see that it's phase 2 metaphase. So the you've already had the homologous pairs lined up. They've already split apart. So you've had the maternal chromosomes and the paternal chromosomes. They've been sort of shuffled, and then they've moved to separate sides. This is phase 2 of meiosis. And you can see that each cell that is resulting is going to be different. Okay, so if we move on to the next slide. Okay, so this is another multiple choice question. I'll give everyone a little bit of time to answer it, and then we'll go through it.
We'll give about another 10 seconds, so get your results in.
All right, so I can see most people answered D, which is great because that's the right answer. So we'll just quickly go through it for those who got something else. So what you've got to do is go through each of the sections. So pretty much you just have to go through all of the like multiple choice answers because there could be a couple of right answers, like not in the A, B, C, D, but there are multiple things that arginine could be. So if we take up, we'll start with A. So we've got AAU. So the first base that we're choosing, this one's an A. Then we go to the second base up here. We'll do A again. And so we're somewhere, if we take those two, we're somewhere in this box. And so then we take the third base, and that's a U. So taking this letter, and so that would be this. So that that's not arginine. So we know that it can't be A. So if we did B then next, I'll just try and see if I can get rid of those markings, maybe not. Yeah. Okay, so then if we do B next, we've got GAC. So starting on G and then A, so we're in this box here somewhere. And then the last one is C. So that's also not arginine. The next one we've got GCC. So take G again, C up here, this box somewhere, and then whichever one is the third one, it doesn't actually matter because they're all alanine. And that's also not arginine. So by default, it should be D, but we'll just check it. So just quickly get rid of these markings. So the start off with C here, G up here. So we're landing somewhere in this box. And we know that it actually doesn't matter what the third base is, but it's G. So that gives us arginine as our amino acid. So yeah, good job to everyone who got that right. Okay, we'll move on to our next question. See if I can, okay, go to the next one. All right, another multiple choice. I'll give you
Abby,
all
just before we do this one, could you go back a slide and explain the difference between the coding and template strands? Is that relevant to that question or not?
It's not. So it's actually just asking for, so each of the amino acids, oh no, sorry, I don't know what happened there. I'll just share it again. So each of the template strands are, sorry, I don't know what's going on. The coding strand is the one that is the same sequence as the amino acid, and the template strand is the one of DNA that is used to create the mRNA strand. So it ends up being that because you know how they use complementary base pairing. So if they had the coding strand and then the template, sorry, you have the coding strand, and then the template strand is the opposite one. So when they make the mRNA strand off the template strand, it's actually going to have the same sequence as the coding strand. So that does, I'm not sure if that makes sense, but I'm just going to try and get that thing back up. I don't know how it's disappeared. It's just kicking me out. Okay, hold up. Sorry. Okay, I'm really sorry. Here we go. Let's try it again. Is that, need to show this.
Yes, that's back up now.
Okay, perfect. Okay, sorry, I don't know what happened there. But hopefully that answered your question. I'm not sure if I was trying to concentrate on both things. But so yeah, this one's just, it's not really asking, doesn't really need to know about the coding and template strands for this question, but yeah. Okay, so the next question, we'll give everyone a bit of time to answer this one.
Okay, we've got quite a few responses, so we're just going to give it another few seconds so that everyone can get their answers in.
Perfect. Okay, so yeah, most of you answered with B, and that's right. A couple of you said A and a couple said the other ones, but we'll just go through it and we'll have a look. So for this type of question, you're going to need to do a Punnett square. So just to start off with though, we need to figure out what the genotypes are of sort of each of the different people. So it says Susan is not colorblind, but her father is. And so it's told us that it's a sex-linked recessive trait. Oh no, sorry, it wasn't on my pen. Okay, told us it was a sex-linked recessive trait, which means that, oh, it's red, oh well. It's going to be, if we just use the letter B, so if it's recessive, it means that the affected allele is going to be lowercase. And so her father would have the genotype of lowercase b and a Y. And then Susan is not colorblind. So it means that she has at least one of the dominant alleles, because that's the only way her phenotype would be not colorblind. But it says that, so we know her father's allele is colorblind. So she has to have received one of her X chromosomes from her father. So she's got one other chromosome which is affected. Okay, so then it says Susan is married to James, who is also not colorblind. So the only way that James could be not colorblind is if he has the dominant allele. And then he would also have a Y chromosome. And so it says Susan and James are expecting twins, a boy and a girl. So for this, we'll just make our Punnett square using Susan and James. So we'll put Susan up the top. And we'll make our Punnett square. We've got, oh no, hopefully I didn't delete that. Okay, good. Okay, and then we've got James over here. Okay, and then if we just do our thing, we've got XB, XB. And so you can see the top two ones, these are both girls because they've both got the XX chromosomes, and then these ones are the boys. So if we're looking at the top ones for the girls, you can see that both of them have the dominant allele in them. They've both got the capital B, which means that since it's dominant and it's not affected, they will, neither of them will have the colorblind phenotype. So we know we're looking at, oh my goodness, it keeps jumping. No, we're looking at one of these two answers. And then if we look at the boys, we've got capital B for one of them and lowercase b for the other one. So the lowercase b we said before is the colorblind allele, which means that this boy will be colorblind, and this one will not. So it's got a 50-50% chance of colorblind. So the answer is B here. Hopefully everyone gets that one. And we'll move on to our next question.
Okay, we've got a few responses. We'll just give you about another 10 seconds. So get your answer in if you want to participate.
Okay, yep, looks good. Most of you answered with B. So we'll just quickly go through it. So it says that the child has a recessive sex-linked condition. So starting off, we can sort of look at A, and we can say, we know that for a girl to get a sex-linked recessive condition, it means that both her mother and father have to have the condition on their X chromosomes. And so we know that if the father had the condition, and so it's recessive, if he had it, he would also be colored in. But he's not in this one, so it can't be A. And we know that, so for B is our answer. So it's got recessive sex-linked condition. So the reason that the boy here may has the condition is because it's likely that the mother has one dominant and one recessive chromosome. And so she's passed on this one here, and then the father, he's got capital and that, and he's passed on the Y over that way. And then the daughter has got the capital B from both of them. Okay, so B, it keeps doing that. B is our answer. Oh my goodness. Okay, B is our answer. And then for C, we know that it technically, this pedigree works because it could have the recessive here, but it said in the question that it's a child that has a condition, and this is displaying the parent. So it's not C, and it's not D for the same reasons in A. The father would have to have the condition if the daughter had the condition. Yeah.
So Abby, just clarifying, this is from the first module?
Yeah, this is all the module 5, which is hereditary. Okay. And we'll go on to the next one. This one's relatively similar, but sort of just opposite. So they've given you the pedigree, and then you have to try and figure out what type of characteristic it is.
We'll give this another couple of seconds. Just get your answers in.
Okay, most of you said D. That's great. We'll just quickly go through it. Okay, so just looking at it, we have over here, you can see that we've got parents here, and so unaffected parents and affected children. So immediately we're sort of thinking that this is a recessive condition, because the only way that someone could not display it but still carry it to pass it on to their children is if they have a dominant sort of a dominant genotype, dominant and a recessive allele. And so that is the only way. They would both have to be heterozygous. Oh, sorry, that's a why? Yeah, no, yeah, okay. Because we don't know if it's sex-linked or not yet. But the other thing is we've got the fact that a lot more women, a lot more sort of females are affected than males. And so that is a good idea, a good indicator that it's not sex-linked. But sort of just to confirm, we have we've got sort of, yeah, I mean, that's sort of just all you really need. It says the most likely type, so you can sort of just take it from that. So yeah, the answer is, so it's not sex-linked and it's recessive. So the answer is D. Okay, and then we'll go to the next question, which is, we're going off multiple choice questions now. There's actually, so hopefully you guys, so just the part one, part A shouldn't take too long. It's just a straight up content question. And then if you don't want to write out the full procedure for part B, maybe just dot point sort of what you might need to include in your answer. And then I'm not sure, Emma might put up a poll just when you're done. Maybe I'll just give you sort of a couple of like a minute or so. We're going a bit over time. I think that was mostly my fault with the technical difficulties. How are they going, Emma?
We've got about 10 people who've said they're finished. So we'll give you another couple of seconds. Okay, people are starting to say that they're finished now, so you can go ahead and start it.
Yeah. So the answer to question A, there's not really that much to it. It's asexual reproduction. Reproduction. That's sort of just something you've got to learn. But yeah, there's not too many questions on those sorts of things where you just have to straight rote learn them. They're usually about applying your knowledge. And then, so for part B, I'm not sure sort of what you guys, if you just dot pointed it or if you actually read out the full method, but sort of things to watch out to include. You've got to have mention of your independent variable. And so in this case, that would be temperature. And then you've got to have your dependent variable. And so the dependent variable is the thing that you're measuring. And so this time, so you could measure something like the, like counting the number of yeast cells or something like that. That would be a fungus that it does budding. And so then you've got to have your variables that you're keeping constant. So let's go constant variables. And so you've got to have a constant time limit, a constant sort of quantity that you're including, and then you've got to repeat it. And so the sample answer is this. So they've got their independent variable, you can see they're mentioning the 3 different temperatures. You've got the dependent variable. So the yeast cells is the ones that you're changing, and you measure them. Where did it go? You've got measuring them by counting the yeast cells down here. And then, so you're keeping it the constant variables. So you've got 2 hours for all of them, and your quantity is always the same as well. And then for repetition, you've got 9 test tubes and you're putting 3 in each. So each of the different temperatures are going to have 3 samples, which means you're repeating it for each temperature. And so that is also taken care of. So that's sort of how you'd answer that type of question. Okay, the next one, just sort of see if you can draw out what you think might go in each of the circles.
Okay, we have about 13 people who have finished. So I'll give you another few seconds.
Perfect. Okay, so I might put up the answer now, and hopefully you guys have all sort of made a start on the question. So if I put up the answer, oh, why did it do that? There you go. There's the answer. Okay, so I'm just going to explain it a little bit. So this cell here, you can see that we're sort of in the, we're doing meiosis because it's asking us for the formation of gametes. And so in this one here, we're in metaphase. And so we've already done the crossing over. So you can see how the A's have switched. And so you'd know to draw that because in the next cells, you've got that the A's here have switched and the A's here have like also switched. So you know that you're going to have to switch them in the cell here, but then you can see that the B's haven't switched. So they would be kept the same up here. And then in these 2 cells down here, sort of it makes it easier because they've done it for you on the left. You can sort of copy what they've done, but it's pretty much just separating the sister chromatids. And so you have 4 unique cells all at the end. None of them have the same combination of A's and B's. Yeah, so hopefully everyone got something like that. I'll go on to the next one. Yeah, I'll give you a couple minutes, maybe just sort of think about what you need to write down. I won't probably do quite that many minutes, and then I'll sort of write down maybe some ideas of what you might want to include, and then you can try and expand your answer a little bit.
How many more questions do we have before the break, Abby?
Yeah, sorry, we've got one more question after this.
Awesome. And just we've been getting a lot of messages direct to the host and panelists about getting Abby as a private tutor. So we'll put a QR code up during the break, but I've also just sent a link through so you can click on her profile and check that out. She's far too modest to say that herself, so that's why I'm saying it.
Okay, I'm not sure how far everyone is along this question, but just sort of writing down some things you might want to include. So defining things is always very important in these longer questions. So we've got to define protein synthesis, and then so you've also got to describe what is happening with protein synthesis. So describe. So you want to sort of just sort of do an overview, but still be detailed in your explanation of what's happening, maybe maximum like 2 sentences. And then for explain questions, you need to pay attention to the verb. And so explain is asking you to link the 2 sort of processes. And so it's asking you to link protein synthesis and sort of the functions in an organism that make it important. So I'm not sure if those help a little bit. So we'll go link.
In the chat, we've got a question about, is this meiosis 1 or 2?
In the previous question?
I think so. Sorry.
Yeah, so the previous question, so I'll just leave this one up so people can keep writing it. But for the previous question, for the diagram, it included both phases of meiosis. So the first one, so you can see it had sort of the first division. So we did the cell just before the first division, and then it had the second division as well. So it had both phases of meiosis in that whole diagram. Yeah.
Okay, awesome. Thank you. It's looking like most people are starting to wrap up. So if you want to give it a go to explain.
So I'll just put up the answer. So I'll just quickly go through it. So first sentence, we've got a definition of protein synthesis. And then we've got sort of describing what happens in protein synthesis. So we've got the first sort of like part of protein synthesis, which is like transcription. And then we've got a second sentence about the second step, which is translation. And that ends over here. And then this whole part down here is linking it back to sort of why protein synthesis is important. So you've got so we've got talking about sort of why enzymes are needed in the body, so chemical reactions. We've got hemoglobin, and that's used for respiration. And then antibodies as well are used for fighting diseases. And so you've got sort of like 3 examples of why protein synthesis is important. And you've explained what protein synthesis is, and you've defined it. And so you can also do a linking sentence at the end just to sort of wrap up all your ideas. But if you're short for time, it's not completely necessary. It just sort of makes what your whole argument is a bit clearer.
And attendees just asking if you could mention translation as well in this answer.
So there is a sentence here, this one to here. This one's about translation. And so, it's good to sort of go into it. You don't have to do extremely detailed, but try and put as much information into the sentence as possible. So you can use brackets. They're very helpful. I'm not sure if that was your question, but
and then someone else has also asked if you need an example.
For this question, you would need to include a couple of examples, just showing sort of why protein synthesis is important. So you've got 3 examples down here. So you've got enzymes, hemoglobin, and antibodies. You probably could get by with just doing 2 examples, but it's always safer to go for more if you can think of them. If not, just move on and you can come back to the question if you can think of things later. So I'll move on to the next question. And this one's only worth 3 marks, so it should be hopefully a little bit quicker. If you're struggling, just think of the word compare. So you can always use tables for compares. That's sort of the easiest way to answer these types of questions, especially if they're only worth sort of 3 or 4 marks. Compare is, you can do tables. Okay, I'm not too sure how many people have finished.
Got about 10 people saying they've finished, so we'll just give it another few seconds.
I'm just conscious of the time.
If you want to start answering it, people can follow along.
Yeah, okay, might do that. So I'll put up the, we'll just go back. Oh, sorry, put up, there you go. There is sort of the answer. Since it's only worth 3 marks, you probably don't have to have all 3 of these and the difference, but at least 2 similarities you'd need. So we've got changes to the amount or structure of DNA in an organism. So that's the similarity. You've also got, you can have the mutation could be changes to a single gene or to the whole chromosome. And then there's the 3 types of mutations, which can be beneficial, harmful, or neutral. So all of those 3 things occur in mutations, whether it's germline or somatic cells. And then the main difference pretty much is that the germline mutations, since they're occurring in the gametes, they're passed on to the offspring, whereas the somatic mutations are not passed on to the offspring. So they just occur sort of like, I don't know, in your arm or something like that. And so no offspring are affected by that. Yeah. Hopefully that sort of makes sense to everyone. And we'll just go into a break now, just for a couple of minutes, and then we'll get started on the next module.
So we've got a question asking, how do we know which questions we need to do the define, describe, etc., and which we can just jump into?
So I find for the longer responses, sort of anything above like 5 marks, I would typically try and just define sort of the important terms in the question. So that question back there was about protein synthesis. So I would, define protein synthesis. It's not necessarily define, describe, link that you have to do. So that process is good for explain questions. And it's also good for other types of questions as well, but you don't have to do it strictly in that order. And it's sort of just a good way of including everything that you need to do in that one question. So you can sort of jump into it if you want to, but you should hopefully be including those things in your response anyway. It's just sort of a logical way of setting it out so that the marker can sort of see where you're going with all your ideas. Hopefully that answers that.
Okay, the next question is, do germline mutations occur during fertilization?
So germline mutations occur in meiosis. So when the gametes are actually being formed, but fertilization is more just the process of the egg and the sperm coming together. And so no, pretty much in short, the mutations occur, the germline mutations occur during meiosis, and fertilization is sort of a separate process, which so the egg would already have mutations in it in that case.
Okay, another question we've got about germline mutations is, could another difference be between them be this and somatic mutations be the gamete cells for germline mutations? Actually, can you just read that, because I don't know if anyone else can tell. I didn't do bio. I don't know what this is. I'm a law student.
Yeah, I'm just sorry. I'm just gonna try and see if I can. It's doing this again.
Okay, I'll try and read it out again. Could another difference between germline mutations and somatic mutations be gamete cells for germline mutations and body cells for somatic mutations?
Yes, that's another difference. I would say that it's good to probably put in the fact that they're, because the question was asking specifically, if I just go back, it's asking specifically about the effects of germline and somatic mutations. So it'd be good to include the fact that sort of in the long term, the germline mutations would affect offspring and the somatic mutations would not affect the offspring, but you can definitely also include the fact like where they occur, but it's just not quite so relevant to the question. Yeah.
Okay. Last question about germline mutations is germline mutation a mutation of in the gamete or the sex organ that passes on to the gamete?
Yeah, so it's a mutation in the actual gamete. So it's something, so the germline just means it's something in the gametes that affects the sort of like the line of offspring. I think that's sort of like how I think about it. Yeah, and then if it was a mutation in the organ that produces the gametes, that could potentially lead to mutations in the gametes as well, but the actual mutation in the organ would be a somatic mutation, and then the consequent mutations in the gametes would be germline mutations.
Okay, and so enough about germline mutations because I don't get it at all. But the next questions are about what study methods did you use to remember all of the content?
Okay, yeah, there is a lot of content. So I wrote notes pretty much before, like sort of at the end of each topic that I did, I wrote pretty detailed notes. And then sort of coming closer to exams, I would sort of summarize those notes more, but in like a different way. So I'd do like sort of a mind map or something, but trying not to use those notes. So I would write down, I don't know, like the kidney or something, and try and write down as much as I could remember about that. Also just practice questions. That really helps securing everything that you're meant to like know about, because it sort of just throws questions like from all the different modules, and it'll combine modules. That's really helpful. I find that if you're struggling to sort of remember something, explaining it to somebody else, get your mom or your dad, siblings, pets, just like talk to them about it, and it'll like stick in your head much better if you can, yeah, just talk to someone else about it. Those are the main things. Just like for me at least, just writing it down, that just helps a lot to keep it in my brain. Drawing diagrams, yeah, those are my main sort of tips for that.
Just being cautious of time, there are a couple more questions, but we'll do them in the next break. Also, if you are, yeah. Abby has absolutely stunning, beautiful notes. So if you want to get access to those, she will be doing group tutoring later in the week. Okay. If we go on to the next module, that would be great.
Yeah, that sounds good. Okay, perfect. So multiple choice question to start off with, and Emma will put up a poll and we'll see how we go.
Babs, your screen isn't shared.
Oh, it's not? Okay, my bad. Sorry. Okay, totally thought it was. Let's try that again.
Okay, yeah, you're up now.
There we go. Sorry.
Just while you're answering this, being cautious of time, would you be able to, if you have a preference, send to the chat, not the Q&A, but the chat, if you would prefer more multiple choice or more of the longer answers, just because we are going a bit over. That's all.
Sorry.
No worries. You've had lots of questions, but yeah, I think we've got quite a few answers to this, so you can.
Cool. We'll get into it. Okay. Pretty much everyone responded with C, so that's great to see. So yeah, so hopefully everyone understands this. Oh goodness, I did it again. Technology seems to not be my strong point. Okay, let's go pen. Okay, so yeah, answer is C. And so we'll just run through the other responses quickly. So mutations don't often occur in RNA because so like the mutations occur in the DNA, and then the RNA is coded off the DNA. So that's wouldn't really happen. And then if the mutation occurs in the protein coding region, that's when it would affect the phenotype. But the question's asking for no noticeable change. So if it occurs in a non-coding region, then that is when it wouldn't change the phenotype. And then the last one, it did not affect the DNA sequence of any gametes. This one is like possible, but like the question is asking about the person itself. Oh, sorry, no, I didn't read the question properly. It's germline mutation. So if the germline mutation, sort of like the definition of a germline mutation is that it is of the gametes. So this one, yeah, it affected the gametes. So this one's wrong because of this sort of idea up here. Yeah, we'll go on to the next question.
Okay, awesome. From what I've been getting in the chat, lots of people are thinking if you could focus a bit more on long answers. So maybe just choose one or multiple choice for this section.
Yeah, that's fine. We'll just do this one. We'll skip the next one. And then we'll go into the long responses. That's fine.
Also, for people asking questions, we have not forgot about you. There'll be another like break soon where we'll get to them. So we will get answers. Don't worry. Okay, people are responding pretty quickly to this one, so we won't give you too much longer. Yeah, okay, we've got 80% have responded now, so we'll just get going.
Okay, wonderful. Okay, yeah, so most people answered B, which is good to see. I'll just get rid of that so I can, where did my mouse go? Sorry. Okay, so the purpose of cloning in agriculture. So the first one, it's saying increasing the frequency of recessive traits. It may do that if you were cloning organisms with recessive traits, but that's sort of not the purpose of cloning. The purpose is B, so you're wanting to sort of keep favorable traits. So I don't know, for example, cows that produce lots of milk, you're trying to, well, you wouldn't really be cloning cows, but sort of wheat or something that produces a lot of good wheat. You want to try and keep that so it's going to be producing the same amount. So the answer is B, but then, yeah, so genetic variability, that's sort of something that cloning doesn't do. So cloning is something where you're like replicating the exact same DNA in the next organism. So it would be decreasing genetic variability. And then increasing combinations of alleles, that's also, it's same thing as the genetic variability. It's sort of changing that instead. It's not increasing the alleles, the combination of alleles, it's just keeping them exactly the same as they are. Okay, so we'll go on to the next, we'll skip this one and go on to the next sort of longer answer response.
The slides will be sent to you in an email after the lecture, so don't worry if you wanted to do that question, you'll be able to see it later. Okay, we'll give you a little bit longer because we haven't got too many responses yet.
This question is just quite similar to the one sort of we were doing before. So I don't know, I might sort of get into it just so we can move on to sort of like the more longer response type questions that are typical, that I think you sort of were wanting to focus on. I'll just plug this in. Okay, so I'll just start. You can still keep answering it yourself if you want to. So the somatic mutation location, so that's in body cells. The germline mutations is in the sex cells. The effect on the offspring, so it's the somatic cells, so it's not passed on to the offspring. Whereas the germline mutations passed on to the offspring. And for an example, so you can just say sort of like a skin cell leading to cancer or something like that. You might want to say sort of like skin cell DNA or something. You could just put that in little brackets, DNA. And then germline mutations. So if you had sort of a mutation, you could say in the sex cell mutation of hemophilia, in the offspring. So you've got sort of some examples. There's lots of different examples. So if you had something else, that's probably fine as well. Okay, we'll get on to the next question. So there's sort of two parts to this one. Yeah, just give it a go answering them. I'll give you a bit of time for both of them, and then I'll sort of do A, and you might be able to finish writing B as I'm doing A as well if you need.
Okay, we're not getting too many responses yet, so I think people are still working. But if you want to give them a hint, maybe.
Yeah, okay. So for the first one, sorry, I'll just get this. For the first one, it'd be good sort of to talk about, you need sort of like link a reproductive technology to the changes in the genetic, that's, yeah, I've just said the question again. Okay, so the reproductive technology that they were sort of talking about is the pre-fertilization sort of one. So you need to sort of mention that in your response. And then you might also sort of want to talk about how this would affect the population. So if I get up an answer, yeah, so it's talking about how the offspring, the effect on the offspring, and yeah, and it's talking sort of like a long-term effects. So yeah, it's saying that the gene will be selected against, because if you're altering it before you're having children, then the allele will become less frequent in the population. So yeah, it's saying fewer people will suffer from breast cancer, and therefore because fewer people will have the gene. Hopefully that one's not too complicated. And so then the next response, it's sort of talking, so this first bit here, it's talking sort of just general benefits to genetic technologies, so reducing the number of people suffering from certain medical conditions. And then, yeah, it's saying, so because of that, what will be the positive impacts on society? So it's increasing their contribution to society and reducing the medical costs of treatment. So that's this bit along here. But then it's also going into the fact that there are sort of negative effects on evolution and ethical issues behind it. So it's talking about sort of insurance companies here, like sort of the economic side of things. Yeah, and then in this paragraph, it's more going into the ethics behind it and sort of problems with using this technology sort of extensively. Yep. Okay, we'll go on to the next question. This one's sort of a longer one as well. So, yeah. Oh, that one has the answer on it. Oh, well, we'll just go through the answer. Sorry, I don't know why the answer's on it already. Okay, so maybe you can sort of try not looking at the answer, but we'll just sort of go through it. So it's starting off talking about evolution. Well, it's not starting off talking about it, but if looking at the question, it's asking you to talk about evolution and provide possible reasons. So those are the main sort of things. And then also, since they've provided you with the graph, you need to be specifically referencing the graph. So a couple of marks will come from literally just talking about, oh, there's a higher frequency in these parts of the world versus these parts of the world, and just using the map. You can try using the key. It's a little bit sort of a vague key, but you can sort of say, I don't know, Australia is sort of about 40% or something like that, sort of approximate if you want to. This answer hasn't done that, but you can if you would like to sort of just make it really obvious that you're referencing the map. It's got a couple of reasons. So yeah, the question was asking to provide possible reasons. So where are we? We've got so milk made available. So less likely to have had milk available, that's a possible reason, which means the mutation would not become more common in the population. So that's justifying it sort of for the like lower percentage areas. And then we've got sort of, yeah, presence of milk in the diet is sort of the main reason that it starts off talking about. And yeah, it's talking about sort of, where are we? We're talking about mutations here. And then in that sentence, it's sort of talking about how the mutations of the lactose gene is really important for digesting lactose. Yeah, hopefully all of that. I'm not sure if there's any questions about that question.
We have one question just asking about how in detail do you need to go about the action potentials?
The action potentials?
I don't know. Is that relevant to this? Maybe not.
No, not really.
Okay,
yeah,
we'll come back to it in question time.
Yeah, okay. Cool. All right. Okay, we'll go on to the next question. This one's another like table again, but sort of on different different. Oh my goodness, why are all of the okay, wait, let's see. Okay, yeah, I don't know why all the answers are coming up before. Maybe I'll need to edit the slides a little bit. Sorry. But anyway, so we've got artificial insemination and IVF. Yeah, so it's asking for a compare again. So I've used a table, we've got the similarities and the differences. I think it'd probably be better to have at least one more similarity or difference. So you could say another difference is that you could say artificial insemination is you don't need to extract the egg, but for IVF you need to extract the egg. Because yeah, as we've said before, sort of we'll just extend this table down a little bit. There we go. Yeah, so fertilization for IVF occurs outside of the body, and then the zygote, which is like sort of replicated a couple of times. There's sort of a bundle of cells that's inserted into the female, and then it grows from there inside the female. And then the artificial insemination that occurs inside the body, so that it's just that the sperm is inserted into the female. Yeah, those are the examples for there. We'll go into question time. Sorry, just running behind and conscious of time. Hopefully that is sort of makes sense for everyone.
Okay, so we have some questions about exam technique. So the first one will be what to do in the reading time.
Yeah, so reading time, it's only 5 minutes. So I would quickly sort of skim the multiple choice questions. Don't need to go through them in a great detail, because you'll be doing them first, and so you can come back to them soon. But then sort of just read through each of the questions. Just sort of think about them as you read through them. Think, oh yeah, that's not too hard, I can answer that. Or maybe it's a question you're like, oh no, but like don't get freaked out by it. You'll be able to think about it whilst you're doing the rest of the paper. And hopefully by the time you come to it, you sort of thought about your ideas a little bit. Make sure you read the questions at the end of the paper because they're a little bit longer, and you sort of want to be able to, in the reading time, you can sort of go to the end responses and sort of like think about how you might answer them. You want to sort of, I don't know. And then if you've sort of gone through the whole paper, read all the questions, you can come back to the beginning, do the multiple choice questions in your head a little bit. And then, I don't know, 5 minutes goes pretty quickly. So you probably won't have that much more time than that. Yeah.
Okay, yeah. And then one question that's not in the Q&A but was sent as a private message. When answering questions that are long, so for 6 or 7 marks, for example, a justify or evaluate question, what do you need to include to get all 6 or 7 marks?
Okay, so it depends on the question. For those ones, there'll probably be 1 or 2 things you've got to define in the question. So you'll start off with them. They'll sort of just be the keywords, the processes, something like that. And then sort of discuss or justification questions, you need to have, so for discuss questions, you've got to have sort of positives and negatives. You're sort of discussing, or maybe like similarities and differences. You're sort of talking about both point of views. So if it was a 6 or 7 mark question, you'd probably want to do 2 or 3 for each, plus your definitions at the beginning. For justify questions, you need to have like a judgment statement. So you need to actually be making a point. You've got to have your definitions. I think we'll go into a couple more. We've got a couple more longer response questions coming up. But yeah, you need to be able to, what was I talking about? Justify. So talking with you've got to have a judgment and make a point, and then justify it with examples. And
so do you have like a specific structure that you follow? Or not really?
There's sort of slightly different for each question, but you would sort of want to generally either define or describe the main terms in the question. And then depending on the verb that's used in the question, so if it's like justify, you want to sort of use examples, and you want to, I don't know, it depends on the question, but like talk about like the processes involved, or something like that. And then discuss, you'd do similarities and differences. Evaluate, you'd also have like a judgment, that sort of thing. It does depend on the question. You want to sort of put as much as you can sort of think of that relates to the question into it. Try and use examples where you can, that sort of thing. Yeah.
Okay, awesome. So more about the exam, we'll just go through a couple more in this break, but about if you haven't memorized all of the content but understand most of it, what would you recommend? Doing some memorization or doing question practice?
I reckon if there's like only like sort of a couple of things, so understanding the content's great because it means you can sort of actually answer most questions that they'll throw at you. There's not that many questions that are sort of straight rote learn type answers. They'll only be worth like sort of a couple of marks usually. But if there's particular things, maybe write some flashcards. You might want to try and use them to help you really remember, like sort of just straight like rote learn the content. Practice questions are definitely good because you see what you can remember and what you can't remember. But if you're struggling, just to sort of remember the facts, probably, I don't know, maybe sort of even just like reading through your notes and then covering it up, repeating it back to yourself, writing flashcards. Again, just even talking to someone else about it, just saying, this is this, like, I don't know, you can talk to yourself even, it doesn't really matter.
Abby does it a lot when she's studying. Anyway, the last question we're going to get through before we go on to the next module is, should you try and attack the last questions of the paper first, or do you want to do it in order of the paper?
I think both are valid. I always did it in order. It's usually the case that the earlier questions are sort of worth less marks and a little bit sort of easier. And so it sort of gets you in the zone, especially like I liked starting with the multiple choice as well. So you start with them, they're sort of
A bit
like
of
not,
a warm-up.
yeah, a warm up, exactly. And so you sort of get in the zone a little bit. And then I just worked through the paper systematically. But if I don't know, there were questions that I'm like, oh, I'm not too sure about this one or something like that. And so you can come back to it at the end. And then usually by that stage, you're like, oh my goodness, I was being silly. I was freaking out over nothing. And then you can answer it when you get there. And so, yeah, I'll just work through it normally. Hopefully at the end, you've got time to come back and go through it again. Yeah.
Okay, awesome. There are some more questions, but we will have another break soon. So you haven't been ignored, that we're going to get to them. But if we can go through the next content block.
Yeah, cool. Okay, so we're starting off with a multiple choice question again.
Okay, we're getting quite a few responses, so I'll just give you another couple of seconds, get your answers in if you want to.
Okay, cool. Most people answered A, which is great. We'll just work through it. So yeah, so the answer is A. So that means that antibiotic W would be sort of the best one to use. So you can see in both of them, it's pretty much consistently effective against bacteria A and bacteria B. And so that's very good, because if you don't know what type of bacteria you're treating, then that antibiotic would be great for treating both of them. You can see that Z is sort of not great in either of them, so you probably wouldn't want to choose that one, because it's sort of much smaller. It's not great at sort of fighting off the bacteria, and same over here. Y is also consistent, but it's sort of just less than W, so not quite as great. And then X, you can see here, it is really good at one of the bacteria, but the other one, yeah, really not great. So if the patient had bacteria B, then yeah, well, it's got both of them, so it wouldn't be good at fighting off bacteria B. So you'd want to go with the one that's sort of consistently good at both of them, which is W. Okay, I'll go on to oh my goodness, it cut off again. Don't know why it keeps doing that. Okay, just quickly go back.
Whilst you're doing that, this should be a pretty easy question to answer while you're doing it. So when you did your bio HSC exam, did you have a lot of time to spare?
I did. I think I finished at least, well, I don't know, it's different for everyone, but I think I had sort of about an hour left over at the end. I sort of, when I'm in exams, I get really stressed, so I write as fast as I can. I imagine that I have no time left, and so I'm doing it as quickly as I can. Oh no, just, yeah, okay, and then we'll go, sorry. And so I had quite a bit of time left over, but it's obviously different for everyone. If you're going through the paper in more detail and answering every question perfectly initially, then you'll probably be taking longer, and then you might sort of just finish with maybe 20 minutes to spare to go back and check. It depends how you like doing exams. By this stage, you've probably done quite a few, so whatever has been working best for you would probably be best. Try and answer the questions in as much detail as you can, but then if just do it as fast as you can as well, because, you know, you are under exam conditions. So you want to be answering everything quickly, so you can have time to come back to it if you need. Yeah. Okay, on to the next one. Okay.
Well, whilst people are answering this, someone's asked if that made you make more mistakes by rushing.
Yeah, so it's definitely something to watch out for. You've got to make sure you're answering every question. Like, I definitely had to just sort of like slow down and be like, take a couple of deep breaths. I think that when you come back through it, like I finished the paper knowing that I would have to go back through it and watch out for things. And it's not necessarily silly mistakes that you got to watch out for, just making sure you've answered the question in as much detail as you can. So when I'm going back through and checking it, like I'm just adding sentences every now and then, things that I realized I could have included. Yeah, it's definitely not trying to rush through it and just get to the end as fast as possible, but you are trying to, I don't know, write quickly. If you know what you're saying, may as well write quickly, that sort of thing. Yeah.
Okay, if you want to get your answers in, just so we can make sure we get to those longer questions that everyone's been asking for, I'll give you another few seconds.
I might skip the next multiple choice question just because it's quite similar. There's still another one after it though. So, and then we'll get to the longer responses just because I think that's what people want. Okay, so yep, most people answered A. That's great to see. Okay, with, oh, I need to get to my, sorry. Okay, this one, oh no, I've gone back, there we go. With this one, so it's asking, you know, yeah, what can be inferred from the scientist's discovery? So the, we'll just read through it. So the melanomas are characterized by uncontrolled cell division caused by mutations that continue to occur once the tumor has developed. So the vaccines that produced antigens extracted from the patient's own melanoma cells were useful in treating the melanoma. So then it's saying that the vaccine stimulated an immune response. So that's indicating that there's something sort of unique about these cancer cells. So, which is sort of what answer A is. So they have their own antigens, which can be used to specifically target the cancer cells once they've been, yeah, so the body can specifically fight these cancer cells using the antigens on them. Yeah, so the other one, that's sort of just like the opposite. The melanoma patient has a dysfunctional immune system. That's not true, because we've seen that the immune response can be triggered by a vaccine. So their immune system is functioning. And that's the same for this one. So the body was mounting an immune response against the vaccine, which is the cancer cells, against the cancer cells. So that's that one. We'll skip the next one because it's pretty similar. And then we'll go to this one and then our longer responses.
Okay, we're starting to get a few answers, so people, get your answers in if you want to interact. We've got another few seconds.
Okay, yeah, so it was a little bit split because there are sort of two answers that look kind of like they could both be right. So for this one, you're trying to find the drug that has low toxicity and high inhibition. So because you don't want it to be toxic to the person, but you also want it to be very good against the virus. So looking at the different ones, you have to be just careful a little bit of this one because their scale on this axis is slightly different, but everything else is pretty similar. So just looking at it, we've got the toxicity levels of all of them are relatively similar at because they're asking for 1 micromole per liter dosage. So toxicity levels are all relatively similar. So let's now look at inhibition and then see if that can differentiate some of them. So for A, the inhibition's a little bit everywhere, for like it goes kind of up and down. At 1, it's, according to the graph, it's here, which is not great because that's 0. For this one, using the graph, this point here, it's sort of between 30 and 60, about here. For parts, for like option C, you can see that the graph indicates that it's sort of about here. If you followed up the line, about here, which is 60. And then the next one, part D, that's sort of only here, which is, I don't know, maybe 15. So the right answer is C, because it has the, and then if you were sort of, it's got the highest inhibition. And then if you're comparing it to B, which is the one that's sort of closest to it, they have very similar toxicity levels, which is sort of just below 0 here and just below 0 here. So you'd choose option C because it's got slightly better inhibition. Okay, we'll go to causes of infectious disease.
Okay, just while people are answering this, so we can address as many questions as possible, how would you tackle data analysis questions because from the new syllabus, there seems to be a lot of data related questions?
Yeah, so there are more questions. Okay, so how do I tackle them? That's sort of, it depends a lot on the question. Usually for the data questions, they're wanting, if you're having to write a response to them, they'll want you to very specifically reference the data. So actually use the numbers in the data that they've provided, use the, I don't know, the names of the bacteria that they've provided, you have to make it really obvious to the marker. They're probably tired from marking, dozens of papers every day. So make it just really obvious. Look, I've used the data you've given me. And then it depends on the question, but if it's asking you to sort of explain a trend or something, you can say that, I don't know, it starts low, increases to a peak, and then declines again. And then you can say, maybe suggest possible reasons for the increase, for the peak, and then for the decline. You have to sort of point out different parts of the graph, or I don't know, it depends. There's lots of different types of data analysis. They might ask you to draw a graph, make sure you've got your axes, make sure you're labeling everything properly, that sort of thing. Yeah,
that would be my 2 cents as well. I did Chem, and our teachers always marked us down if we didn't explicitly say, like if the question said 3.4 something, you have to say 3.4 something.
Yeah, and make sure I don't know, bio doesn't have that many, like units are still important, not as important as Chem, but if you can include them and if they're provided, then do that.
Okay, we've got about 5 or so people finished, so just like give it a go at starting. And
so I'll just put down, so I'll just start off with sort of things that you sort of would want to include. So it's really quite clear, it's asked you for 2 methods. And so for my example, I've chosen genetic engineering and quarantine restrictions. So those are sort of the 2 ones that I thought of. It's a bit of an unusual question because it's a describe question, and these ones are typically not worth 5 marks, but it is asking for 2 methods. So you're going to have to go into detail for both methods. So for each, I would say that you need to sort of what the method has changed in terms of how the technology works. So I'll just put up my answer. I feel like it'd be easier if I'm sort of explaining what I'm doing. So for genetic engineering, so what it changes is it changes the genetic characteristics, and it can like produce insect resistant crops. So those are the two sort of things that genetic engineering does. And then you also want to link it to the other part of the question, which is like the management of plant diseases. And so that part comes in, and you're saying that it has resulted in less need for insecticides. And so that's sort of how the management of the plant diseases has changed, because you've been able to produce insect resistant crops instead of using the insecticides. And so then for the other one, quarantine restrictions, it's saying that, yeah, the quarantine restrictions has prevented the spread of plant diseases into and around Australia. And then the change in management, so you're talking about both parts. Change in management is you're changing from treating the diseased plants, and you're changing to preventing it instead. So it sort of just makes it easier to control disease in Australia, because instead of having to treat all of the plants, you're just stopping the diseases in the first place. So yeah, those are the two ways that you would get those. Okay, we'll go on to the next question. This one's a little bit longer, so I'll give you a bit more time. Maybe just try answering part A and B first, and then we'll do part C after we've done part A and B.
With the last questions, could you also mention pesticides?
Yes, so pesticides and insecticides, that sort of goes into the first one. It was asking for genetic technologies, which means you needed sort of, no, it wasn't. It was asking, wait, hold on, let me just, what was the question asking? It was asking for, oh yeah, so yeah, you can talk about that. Yeah, sorry. You can talk about pesticides. I was reading the question wrong. So yeah, again, just do part A and B, and then we'll do part C later.
In the interest of time, if you're happy to just get started on us answering it, just react yes.
Yep.
Oh, okay. People are now all reacting yes. They're happy for you to get started.
Okay. Sorry. Sure. Okay. So for this one, I'll pop up the answers for A and B, and then we'll just go through them. If I can click on it properly. Okay. So part A is pretty much just a simple identification question. It's a helper T cell. So yeah, these cells come through and they will sort of activate the B cells using the cytokines, or like interleukin 2, you might have more specifically used. Okay, and then part B, the role of plasma cells as shown in the diagram. So you have to make sure you're using the specific, like, so this one, we're using the specific name of the, what's it called, the bacteria. And that's good. It shows you've actually read the question. You're not just sort of writing down whatever you feel like. Okay, and then so the role of plasma cells, you need to mention antibodies, because these are sort of like the main things that are produced by the B cells. Yep. And then so the role of antibodies there, so they have sort of 3 methods of fighting bacteria. So they clump it together so that the macrophage can destroy it. They bind to the surface of the bacteria, and then they activate the complement system. I don't know why I needed to underline all of them, but there you go. And then you also need to mention, it's already set up here at the top, but yeah, the plasma B cells are specific to that bacteria. Yeah, so it's only 2 marks, but just try and sort of put as much as you can think of that relates to plasma cells, antibodies, how they fight everything, but it don't need to go into like a long-winded explanation, but just sort of, yeah. Okay, so maybe try part C now, and then I'll put up some, yeah, I'll just give it a go on part C and then I'll start answering it soon. Hopefully you can still see the diagram. Sorry if it's too small.
Okay, yeah. Are you ready to give it a go?
Yeah. So for part C, there's a couple of things you need to mention. So it's talking about sort of the immune response, and this graph is more specifically talking about sort of like the different stages of the immune response. So you need to talk about memory cells. And in this case, it's the innate system, so the memory B cells. And then you've got to, so the graph is really specifically asking you to talk about the difference in primary exposure and secondary exposure. So primary versus secondary. Went a bit off the side. And then you also need to make sure that you're specifically talking about both diagrams. And so mention particular parts of both diagrams, because it's really asked you for both of those things. So if we get an answer up, then, so yeah, we've got already, we're talking about specific like receptors. So that's important for your immune response, because once you've learnt the, you've got memory cells specific to those receptors, then you can activate the secondary immune response, and it's much faster. And you can see in the graph as well, the concentration is much greater than your primary immune response. So yeah, primary exposure to bacteria, and then you're saying that you've read the question, so you've got the vaccination here. Yeah, and so you're producing the memory B cells as a part of the immune response. And so, yeah, and so you've got specific mention of like the graph. Yeah, the B cell needs to be activated by the interleukin 2. And so then that's talking about this graph over here, the cytokines. This is, so interleukin 2 is a type of cytokine. Interleukin 2. Okay, and then, yeah, the antibody secreting plasma cells are produced this way. So you're talking about the plasma cells. And then back to the graph, you're saying here that the secondary exposure allows for more antibodies to be produced and in a shorter amount of time. And then, yeah, so memories B cells specific, and then they differentiate into plasma B cells to fight the infection more efficiently. So that's sort of the role of the vaccination. And you're talking about both of the graphs, both the diagrams. Yeah, okay, we'll go on to the next one. This is the last one before the break.
Here's a question just about the last question that you answered, saying, wait, yeah, what does the interleukin 2 do / side token?
Yeah, so that is released by the helper T cell, and it activates sort of, yeah, sort of like activates the B cell to differentiate into the memory B cells and into the plasma B cells. And so that the helper T cell, those cytokines are what actually sort of tells the B cell to like make the memory cells and then produce the plasma cells, which therefore go off onto producing antibodies, which actually fight the infection. So the interleukin-2 is sort of just a messenger that is going from the T cell to the B cell. Does that make sense? I think, hopefully.
I'm sure it did, if I knew anything about biology.
Yeah, I always ask that because usually people reply to me and then I realize that they can't. So hopefully that makes sense.
Someone's asking, what are cytokines and chemokines?
I'm not too sure what chemokines are. I think you only really need to know about the cytokines. And so that, yeah, as what I described before, you just need to sort of know about how they are the sort of messenger between the T cell and the B cell.
Also,
we might just get into
this last time. Yeah, if you want to just get into it, just reply yes.
Yeah, I'll just about to. I feel like this one's only 3 marks, so we might just sort of get into it.
Yeah, responded. So
yeah, so this one pretty much it's 3 marks, so you need to maybe just talk about sort of what antibiotics do and then outline a benefit and a limitation. It's not an overly complex question, so we'll just pop up the, if I can get the answer up, pop up that answer. And then we'll just go through it. So it's, yeah, they've sort of combined the benefit and the definition kind of. So it's saying antibiotics can be used to treat bacterial infections as they inhibit bacterial growth. So you could split that up into 2 sentences and sort of just talk about how antibiotics work in terms of, like they inhibit, like antibiotics are a sort of drug that inhibit bacterial growth. And then you can say a benefit. You can really be very explicit. Just be like, benefit is that they are used to treat bacterial infections and, stop people dying from bacterial infections. And then you can say that limitation of using, of like overly using antibiotics is that it's creating antibiotic resistance in bacteria, and so it's reducing the effectiveness of these antibiotics. Yeah, it's not, yeah, as I said before, it's not an overly complex question. You've got to just try and look at sort of how much you're meant to say based on the marks. That's really useful, just looking at the marks of the question and seeing how much detail you need to go into. Okay, and then we'll just quickly go into break, so if I can answer some more questions if they're coming up.
Okay, this one I have been ignoring since like the first module, but what causes the mutation in germline cells?
Oh, well, it's sort of just the same as mutations in other cells. So it's just a part of meiosis that goes wrong. So it could be in the replication of the DNA. So it could just be mutations that occur in the replication, or there could be problems when there's, I don't know, crossing over problems, or the chromosomes might split out in ways that they're not meant to. So you could get sort of 3 chromosomes in one cell and only 1 in the other cell, and that would cause sort of the typical things. So you might know of like Down syndrome. That's when you have an extra chromosome, so that's how that can occur. Yeah, and so it occurs during meiosis, pretty much, in short.
Okay, our next question would be, could you explain the could you please explain the hormones? I always get confused which we need to know.
The hormones, was that?
Yeah.
So we'll be sort of going into that a little bit in the next module, but okay, it's sort of used in different things. So you're gonna need to know hormones involved with the kidneys. So you've got insulin. I think that's probably the main one. So you got insulin and the glycogen. Those are the sort of the positive and, well, not the used in the negative feedback loops, sort of for an increase and decrease in glucose. Other hormones. So you've got sort of like the ones used for pregnancy. I'm not sure if this is what you're getting at, but you also have so estrogen, progesterone, and you've got HCG,
Yeah, I think the same person has asked like for pregnancy fertilization.
Yeah, okay, sorry. So yeah, you need to know about sort of estrogen, progesterone, HCG, the luteinizing hormone. Sort of, I feel like for studying for that, it'd be great to draw yourself a graph that has sort of the motions of each of the different hormones over pregnancy. So you can see how all of them are influencing the other ones. There's also, if you don't want to draw one, you can just look at graphs on the internet. There's really good ones out there that have sort of all of the hormones in the one graph, and you can see, I don't know, yeah, the different like spikes in the hormones and how it influences the other ones. I think those are the ones I can think of off the top of my head. Yeah.
Okay, and the last one before we just get on to our next content is just, do we have to explain how antibiotics work, like for the question that you were doing before?
Since that question was only 3 marks, you probably don't have to go into it. You can just say the basic antibiotics fight off bacterial infections. If it was a longer question, say if it was worth maybe 5 marks, you may have to sort of go into detail about how antibiotics work. And then I think the question, it said, so the question just said outline. So it just has to be pretty basic. And then it was a benefit and a limitation. But if it was worth sort of 5 marks, the question might be phrased slightly differently. And you could go into sort of 2 benefits and 2 limitations. But yeah, it just depends how much it's worth. For 3 marks, you probably don't have to go into the specifics of how antibiotics work. Yeah.
Okay. So if we go on to our content, in this module, are we doing action potentials? Whoever our friend was who asked that like ages ago?
No, I'm not too sure. I'm just trying to think of, I can't really remember doing action potentials. I'm not sure if that's just, I didn't study that one properly.
Rephrase your question, whoever's wondering that one?
Yeah.
Okay. Let's do maybe one multiple choice. Yeah?
Yeah.
So just choose your favorite one.
I can go into action potentials at the end if you'd like, sort of just after this module. Yeah.
Okay, we've had 10 people answer. Let's get a few more. Give you another few seconds.
Oh my goodness. Annoying. Okay, sorry, I'll just get it back again.
The joys of modern technology.
Yeah, I don't know why it keeps kicking me out. Oh well.
Okay, I'll end the poll here.
There we go. Share screen again. Okay, so it's quite a split response. I'll just get into it. Okay, there we go. Let's close that. And okay, so it's maybe a little bit confusing because oh my goodness, it just kicked me out again. I don't know why it's doing that. Okay, okay, I'm just gonna try and say it from memory. So it may be a little bit confusing because the feedback loop looks like it's sort of going in, like it's an increase in, oh my goodness, stop. Sorry. It looks like it's an increase, but it's actually, if you look at the first part, it's a decrease in temperature. So you need to be thinking about that. And so when you're cold, it's asking, sorry, I'm just, there we go. When you're cold, asking for the possible response to a decrease in temperature. So for this one, it's asking for, okay, so for vasodilation, that would, oh my goodness, that would occur when you are too hot. So the blood vessels would be dilating near the surface and you'd be trying to release as much heat as possible. The reduced metabolic rate, that would also be doing that. And then the vasoconstriction is what you're wanting to, so the vasoconstriction is the answer. You're trying to prevent, I don't know what's going on, trying to prevent the heat from escaping to the surface of your skin. So if you make your blood vessels smaller, then there'd be less blood near the surface of your skin. And then sweating would be for an increase in temperature as well. So we'll go on to the next one so we can get through these.
Will this be our last multiple choice?
There's two more, but we can skip over them if we need to.
I think that would be good. We only have like 15 minutes left.
Yeah, sorry. Let me just see what we've got coming up. Yeah, okay. Yep. Okay, I'm just gonna get into this one. It's relatively sort of similar idea to the previous one. So yeah, most people answered A, which is the brain, so that's great. And so yeah, it's just sort of mostly just remembering that temperature is controlled by the hypothalamus, which is in the brain. That's pretty much all there is to this question. Yeah, so we'll just go on to the next one. We'll just go on to the long responses so that we've got time for all of them. Okay, so everyone have a go at this and we'll go through the answer.
People are saying, yeah, move on, like answer it.
Perfect. Yeah, so it's the main difference between temperature and glucose. So put up the response and it's pretty much just there's sort of like the differences is in the receptor and the sort of system that's used for response. So the receptor for temperature is the hypothalamus and the brain is the pancreas. And then the system that's used, the temperature is the nervous system, and for glucose, it's hormones. So that's pretty much all there is to the question. And then they've just put it together. Okay, next question, this one. So this one's 5 marks and an explain.
Okay, I think people are also just wanting you to start going through it.
That's all good. So for this one, you're wanting to include, it says support your answer with examples. So include at least 2 examples. And then you're needing to link the purpose of educational programs. So you need a link, so how it actually is reducing the incidence. So to start off, you can sort of define what the purpose of them are. So the education programs raise awareness of environmental agents. So this one, they're doing an example for melanoma. So they've put up 2 examples and then they're explaining one of them. Okay, so the UV radiation can cause melanoma and tobacco smoke. They've explained the UV radiation for melanoma, and then they've explained tobacco smoke for lung cancer. I just kind of crossed that out. I didn't mean to. So they're saying that the purpose of the educational programs is to alter behavior in people. And so they reduce the exposure to harmful situations. And then not everyone can avoid the sun, but the program, tells people what strategies they can implement to reduce UV radiation. So they've sort of explained the melanoma one in more detail than lung cancer, and that's okay. I would say if you have time, probably explain lung cancer in a little bit more detail as well, just to make sure you get the full 5 marks. But yeah, this is okay if you just go into detail, but make sure you've got one of them fully expanded. Okay, and then this is our last question. So it's 7 marks, and you want to go into evaluate the effectiveness of renal dialysis in managing the loss of kidney function. If you want to, you can sort of, move on.
We'll just give them another few seconds. A couple of people have said no, not yet.
Oh yeah, that's fine. I was just going to say that if you want to, you can sort of just dot point what you think might need to be included, and then I'll do the same, and then you can expand your answer if you if I've put up stuff that you haven't included yet. Yeah.
Okay.
So
get going.
Yeah. Okay. So for this evaluate question, you're going to want to include a judgment statement. And that's really important because you actually have to take a stance on what you think. It's probably easiest if you put this one at the beginning because it's sort of you're answering the question, but you can put it at the end to sort of summarize if you need to. You can do both if you've got time, but as long as you include it somewhere, that's really important. So what I would do, I would sort of start off with my judgment statement and then define normal kidney function, because the question's asking about sort of managing the loss of it. So you need to define what is normal and why that's sort of needed. So define the normal kidney function. And then you've got to define what renal dialysis is, what it does. So that would include both hemodialysis and peritoneal dialysis. So you've got to go into a little bit about both of them. So I'll just put in renal dialysis. Oh, I spelled that so wrong. Just a minute. D-I-A-L. I'm not the best at spelling. Anyway, and then you've got to actually answer the question in a bit more detail. So you've defined your terms, you've made your judgment, but you need to sort of expand upon your judgment a little bit. So I think it's easier if I sort of just show you what's going on. So if I put up the answer, we've got the judgment statement at the very beginning. So it's saying that it's effective in managing the loss of kidney function, but it wouldn't be an adequate replacement. So it's useful in short term, but not for long term. And then the answer, it goes on to talk about sort of kidney functioning and why sort of normal kidney functioning is used. So that's defining the normal kidney functioning and then defining the renal dialysis. So it's got a sentence just generally about renal dialysis, and then it goes into hemodialysis and peritoneal dialysis in just the one sentence. So you're just trying to sort of outline the main differences between hemodialysis and peritoneal dialysis and sort of just jot down the points that you might need to sort of refer to later. So then it's going on, you want to sort of answer the question, the effectiveness of it. So it's saying renal dialysis filters out the blood passively by moving it past and like, you know, different blood concentrations. And so it's saying that the blood is effectively filtered. And as you've said before, the normal kidney functioning is pretty much just filtering the blood. You're doing what the normal kidney would do. So that's sort of like a positive. And then you've got some negatives coming up. So you're saying the process is not as effective as normal kidney functioning because it's passive. And so therefore it can't do all of the active processes that normal kidneys normally do. And then also an additional disadvantage of hemodialysis is it's not regular. Yeah. So, and then you've got sort of just another sentence summarizing what you've said. So this main part here is going into sort of like the good things that it does and the bad things that it does. But this first bit here is also important in sort of establishing what the renal dialysis does. And because without that, you wouldn't be able to go into the positives and negatives. So yeah, that's how I'd answer that question. And that's sort of all I've got there. Do people have any last questions?
Yeah, so we've got someone asking in the chat, how long should you spend planning your answer before you actually write a response?
Yeah, okay, so I typically think that I would only plan the questions worth sort of 6 or more marks. You can also plan them if it's 5 marks, but it wouldn't sort of need quite as much. For this sort of thing, it doesn't need to take that long to plan it. I would really just probably do the same sort of dot points on the side. I might also put it like judgment, and then also put down sort of positives and then negatives. And that's pretty much all I would do to plan it. And then I would just write sentences based on those things. I don't know, it may be sort of like a minute or 2 per question per like long answer question if you feel like you need it. I definitely think it is useful to plan out these questions. It just gives it a bit more structure and it's easier to read, because once you've done this sort of plan, you can think, oh wait, hold up, I need to put this thing somewhere else. And so you can change it around if you need to. Yeah, I'd say probably a minute or 2 maximum.
Okay, awesome. Someone's also asked, how important is spelling it correctly?
Yeah, it's important if there's something else that it could be mistaken for, like the mistake that I made spelling dialysis there. I kind of noticed right away that it looked wrong, so I fixed it up. But I think if you do spell something wrong, it's not the end of the world, as long as they understand what you're trying to say, and it couldn't be sort of I don't know, misunderstood.
Yeah.
Yeah, because there are a lot of things that are sort of very similar. And so you just don't want it, like, I don't know, if you spelling like mitosis slightly wrong, it could be maybe misunderstood as meiosis, and you don't want to get that wrong.
Okay, awesome. Looking back at the Q&A section, and how much detail should we know the eye functions, refraction, accommodation, something of things, as we learned it during online class?
Okay. Yeah, I do think it's important to know about the different functions of the eye and how each of those, how like problems with each of the different parts could lead to certain like eye problems. So you probably learnt about the reasons you might need, I don't know, glasses or LASIK eye surgery. And so you need to know sort of what goes wrong in those situations. There was a question back here, this multiple choice question. So that's, yeah, as you can see, it's question 11. It's not that far through the paper. So something like that should come relatively easy to you. So just knowing the function of different parts of the eye, and I'm not sure if, did you say ear as well?
No, but go off.
No, okay, I'm just hallucinating. But yeah, probably should know about refraction and accommodation. Yeah, just know sort of like the general principles behind everything, and just be able to briefly explain it in a sentence or 2 should be enough detail.
Okay, the last question that we're going to go through, because the rest of them are all asking what action potentials are. So that's kind of...
Okay, yeah, okay. I can go through action potentials if you want.
Okay, we'll do that after this. But first,
yeah.
Could you explain secretion, reabsorption, and filtration?
Okay, yeah, sure. So this is in the kidney. Why can't I move my... Okay, sorry. So in the kidney, you've got the different tubules and the loop of Henle. And so pretty much reabsorption is where the nutrients are being filtered sort of out of the kidney. And so it's being reabsorbed into the body. It's a little bit confusing. But so that's reabsorption, and then secretion is where in sort of the tubules and stuff, the nutrients are being, well, not nutrients, they'd be waste products, but they're being sort of absorbed into, not absorbed, that's going to get confusing. They're being sort of like pulled into the, I don't know, loop of Henle or one of the tubules. And so that is meaning that it's getting secreted, like because it's in the tubule that's going out. Does that make sense? So the absorption is when it's going out of the kidney tubes into the body, and then secretion is where it's going into the tubules to be like excreted out of the body. Hopefully that makes sense. Was there another term in there?
I don't know. I dismissed it. Well, I answered the question,
Okay.
but the
And
other
action
one. Well, yes. I know we're over time, so if you have to go, please do go. Thank you so much for coming, and thank you, Abby, for answering everything. We'll stay on for a little bit longer. She's done an amazing job. So if you want her as a tutor, that link and the thingy, the QR code will be up. Oh, thank you for saying thank you. That's so sweet. And also, if you will be sending an email after the event, if you want to give any feedback, that'd be awesome, because this is my first series of events that I've run at KIS, and I would love to be able to help people more in the future. So if there was something that we did well or not, just let us know. Okay. Specific eye diseases. Do you need to know them? Yes or no?
No. Oh, maybe just have like an idea of like one that you could talk about if you're specifically asked to. Yeah, you sort of just need to, okay, well, you do need to know sort of like about myopia and hyperopia, and that, and yeah, maybe sort of just like one other one that you could talk about if you're specifically asked. It's unlikely that they're going to be like, explain this specific eye disease, because the HSC is sort of moving away from those types of questions, but you never know. Yeah.
Okay, then let's do action potentials.
Action potentials. Okay.
Saying thank you. You're appreciated, Debbie.
Oh, you're welcome. Okay, so action potentials. These occur. Okay, so this is the movement pretty much of ions in and out of cells to create sort of concentration gradients, and you get sort of movement. You're trying to build, it's called an action potential. You're trying to build a difference in concentration so that messages can be sent sort of along neurons. So you really don't have to know this. I think I remember sort of where you're getting this from now. It's when you go into the nervous system, but you don't have to know this in great detail. But it's pretty much just the start of the neuron will receive a message from the previous one, and there will be sort of an imbalance in ion, like sodium concentration. So the sodium ions will move so that the inside of the cell is more less negatively charged. So it increases in charge, and then that sort of cascades kind of down the whole like axon of the cell, and all of, it means that it pretty much just means that the charge of the cell, it's negative, and then it goes slightly positive, slightly positive, slightly positive, slightly positive, and it moves all the way down the cell. And then it's like fixed up again, sort of it decreases again behind itself. And that's just sort of how the messages are sent along neurons. You don't really need to know this in great detail. It's not a big part of this. I understand that you might be worried because it's a complicated process and they probably just briefly went over it at school, but don't worry about it. It's okay.
And the last thing, I think we talked about this earlier, but like with those longer questions, if you get a specific verb like justify or explain, how would you tackle it? Is there like a structure that you would specifically do?
Well, yes, kind of. There's sort of, I was given a resource from school. I'm just going to see if I can find something similar to it. I think you might even be able to find them on the NESA website. There's usually sort of like an explanation of what each of the verbs mean. I'll just try and see if I can find something. let's have a look and see if this is it.
I love how now that we've finished, your screen isn't dropping out anymore.
Yeah. Wait, so can you, oh no, you're just seeing, what are you seeing right now?
Just a black screen.
Okay, well.
Yeah.
Okay, so you can sort of just look up verbs for answering biology, and then like HSC or NESA or something. There'll be things that can come up, and they'll have sort of like a good, if you just go into images, you can say there's like explain questions. You've got to sort of do features and characteristics. Most of them, I would say, you've got to include defining the terms, and then sort of discuss questions. You've got to talk about positives and negatives. Justify, you've got to, yeah, I don't know, justify, you have to explain why something is the way it is, maybe do a judgment statement. I can't remember what other types of verbs there are.
Yeah, I know at KIS we have a bank of resources that our biology tutors have put together about this sort of stuff. So if you do end up wanting to do tutoring with us, we have like resources that you'll get access to and all of that. So keep that in mind if that would be something that would be helpful for the next couple of weeks.
Yeah, cool.
Okay, awesome. Well, I'm happy to leave the event here. Thank you all so much for coming. And if you want to leave feedback or watch it again, you'll get an email soon. And thank you again, Abby. You did so well.
You're welcome. Okay.